Great, thanks Phil. I will say that it's lovely to be out of the ivory tower.
Hooray!
A lot of fresh air out this way. So, planning on talking to you guys a little bit this evening about the research that I do with designing gels which mimic your brain, and trying to use these gels to get insights into the early stages of dementia. So, before I start, I want to introduce you to someone. So this is my granddad Jim, right? Seen here, the height of mid-90s fashion, right? You can see he's got his sandals on with his socks pulled all the way up. And he's got a very unimpressed looking me on his knee, obviously judging him for his fashion choices. Now, I'm from Perth originally, and my granddad used to live about 3 to 4 hours south of Perth. So me and my little brother got really excited when he would come up to visit us, because he would always bring lots of sweets, right? Iced finger buns, candy, all that sort of stuff. And like a good granddad, he would get us hyped up on sugar and then leave, right? leave us for into the light. Come into the light. So, and he'd leave us for Mum to deal with. So he used to live about 4 hours south of Perth, and I remember one day we got a call from the police saying, look, he's been wandering around a little bit, and this wasn't that unusual, because he used to go on morning walks around the Oval, but it was the evening. And it's a pretty tight-knit community, and one of his neighbours called the police, and the police went up to him and said, hey, where are you supposed to be? Where do you live? And he looked at them and he said, I don't know. So this was kind of the start of when we began to notice that he was declining cognitively. And you know, it got to a stage where he couldn't independently live by himself, and he had to move up to Perth to an aged care facility. They put him on steroids, and that helped for a period of time, and then he became aggressive and had to be taken off the steroids and declined pretty quickly, and he passed away. And it's a sad story, right? But what's even sadder is that this is not a unique occurrence. This happens 10 to 100 times per week in Australia, right? It's quite a regular occurrence. And you can see from the stats that dementia is actually the second biggest cause of death in Australia, behind heart disease. And that's pretty surprising. And it's going to get worse because we have an aging population, right? Some more stats, I mean, 1 in 50 Australians are currently living with dementia. When you change this to people over 65, it's 1 in 10, and over 85, it's 3 in 10. It's 30% of people that currently have dementia. And it's expensive, right? And all of these aged care facilities, the hospital-related admissions, you know, it's $15 billion a year out of the health budget. And dementia is actually an umbrella of diseases, right? It's Alzheimer's disease, which is probably the most well-known one, but you have things like motor neuron disease, PICS, Parkinson's, frontotemporal dementia, and Lewy body dementia. And these are all diseases where you have proteins which misfold within your brain and deposit and cause neuronal death and neurodegeneration irreversibly, right? So I'm just going to very briefly talk about two proteins, two major players in Alzheimer's disease. Right? I don't know if you've heard of them before, but the first one is amyloid, right? So amyloid normally lives within your neurons. These are the cells within your brain, okay? And the amyloid protein is stuck in the cell membrane, and then you have two enzymes normally that come in and cut this protein. So two that come in and do that. And if one enzyme one day decides that it wants to be special and cut outside the lines, then that's when you start to get that essentially bad amyloid, pathogenic amyloid, right? And this amyloid then reaches out to its other amyloid mates and starts recruiting them, right? Yeah, come over here, let's organize an angry mob and a rally, right? And it starts to deposit within someone's brain as these amyloid inclusions here. And that's what starts to develop inflammation. You get inflammation of the brain, you get chemical changes, and you start to get loss of neurons, okay? The other protein is tau. So tau is a bit different. It actually stabilizes things called axons. So if you think about a neuron like a seedling, okay, if you've got a seedling that's buried in the ground, and that's your cell nucleus, all right, and you've got sunshine and water coming down, and that seedling wants to sprout. That's your axon from the neuron coming out, trying to make contact with another neuron to transmit information. What tau does is it stabilizes this axon. It stabilizes the seedling shoot coming out into the sun. But in Alzheimer's, there are chemical changes to this tau protein, which means that it can no longer interact with this axon, and the neuron ends up shriveling up and dying. So the other thing that I want to mention is that we do try and mimic the brain in the lab. We try and mimic the environment of the brain. The reason why this is difficult to do, the reason why it hasn't been done very much is because neurons are very sensitive. Okay, so neurons, the example that I'm going to give you is that I have a cat at home, right? A very cute, gorgeous, destructive cat. And the cat likes to sleep only in one spot, right? The cat will not sleep on the floor. The cat will not sleep on a pile of clothes. The cat will not sleep on a box. The cat likes to sleep right there, right? Neurons are quite similar. They are very sensitive to their environment. They will only grow in a very specific environment. And this is what we try and mimic in the lab. So what we do is we design molecules that self-assemble and try and mimic the environment of the brain. So in your brain, you have neurons floating around, right, and they're supported by what we call the extracellular matrix. And if you're wondering what that kind of looks like, if you've ever been to the gym or you get on a bus and you pull your headphones out of your pocket and they're tangled to all hell and they take you 20 minutes to untangle, that is pretty much what the extracellular matrix of your brain looks like. It's this tangled web of fibers, okay? And that's what we try and kind of recapitulate. So then we put neurons on here. And what we do is we then look at their electrical activity. So I don't know if you're going to be able to see this, but essentially, I'm sorry for the pixelation, but we have pulsing neurons. So these are young neurons on our gels, and they're kind of randomly pulsing. If you can't see it, use your imagination. It's a bit like a magic eye, right? And they're pulsing randomly around. And then once they get a bit more mature, you actually see that they change their pattern of firing. So they go from randomly pulsing to pulsing in sync, right? And this is what happens in your brain. Bang, they all fire together, right? And this is really what we want to work towards with our gels, mimicking the brain. So I guess what we want to do is be able to grow these neurons on our gels for long periods of time, okay? Because even in the most advanced models of Alzheimer's, these things take 3, 4, 5 months to start to develop pathology, start to develop deposits within your brain. Then we're going to culture diseased neurons. So these are neurons that already exhibit signs of Alzheimer's. Then we plan on identifying biomarkers that are associated with Alzheimer's, right? So that at the end of the day, you can go to your doctor 20 years before my granddad went to the doctor, and they can run a test, and they can say, you know, whether you're at increased risk or decreased risk of Alzheimer's based upon, you know, maybe 20, 25 biomarkers that we would have identified in the lab. So just a quick thanks to these guys for the help, these guys for funding, and thanks to all for your attention. Yes.